Hello everyone, welcome to today's webinar. I'm Catherine Dold with CNEN and I'll be moderating today's event. This webinar is titled Variable Pathlength Spectroscopy for Dilution-Free Concentration Measurement in GMP Environments and is sponsored by Repligen. CNEN collaborates with sponsors to identify topics aligned with our mission of providing timely, accurate, and balanced news and analysis for the chemistry enterprise, ensuring value for our CNEN audience. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look for the Help widget at the bottom of the screen or type your query into the Q&A box. If you get disconnected during the webcast, please log back in according to the instructions you received earlier. You're encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. Our speaker will answer them at the end of the presentation, and as your moderator, I'll be posing as many as time permits. Any questions that we are not able to get to in today's broadcast will be answered by email after the event. Please note that CNEN does not endorse any company, products, or services that may be mentioned in the webinar, and that the webinar will be archived at CNEN Online after the live webcast. Our presentation today is sponsored by Repligen. Repligen, a leading life sciences company focused on bioprocessing, has been empowering scientists to effectively address process challenges with innovative solutions since 1981. Their commitment is to inspire progress in bioprocessing by delivering cutting-edge products and flexible solutions. Through the development and commercialization of high-value offerings, they enable their customers to overcome challenges at critical stages of biologic drug production, establishing new standards for efficiency and success in the industry. Our speaker today is Joe Ferriolo, Director, Bioanalytics Applications at Repligen. Joe leads the Bioanalytics Applications team, overseeing the variable Pathlength Spectroscopy solutions for inline and at-line applications. Drawing from over a decade of hands-on experience in analytical applications, Joe plays a pivotal role in enhancing Repligen's customers' success. Joe's leadership ensures successful validations, smooth method development, and the integration of highly accurate process analytical tools into their biologic platforms, addressing critical production stages and bolstering their capabilities in biotechnology. I'll now hand the program over to Joe.
In today's webinar, we're going to highlight the challenges with traditional UV methods. We will show you how our variable pathlength technology works. We will also share some customer-provided information and validation data. And lastly, I'll share some examples of customer publications and discuss some specific variable pathlength applications. At the end of this presentation, we will have a live Q&A session, so feel free to type in your questions at any time throughout the presentation. Over the last 15 years, we have successfully implemented our variable pathlength technology across pharma in both in-process, clinical commercial manufacturing, and QC. These efforts and successes has led Repligen to expand our technology into other modalities such as gene therapy, viral filtration, and plasmid purity. Repligen offers innovative solutions for all the key stages of bioprocessing, from upstream intensification methods using ATF and TFDF through chromatography systems to focus improving on high yield as well as our well-known OPUS columns. We continue to provide systems that improve productivity in the downstream process filtration and formulation systems that are scalable from process development to production. Our comprehensive fluid management offerings allow you to be confident with secure fittings and secure supplies. Repligen's goal is to drive integrated smart systems that will be PAT automated using variable pathlength technology. By integrating our FlowVPX systems into each system, Customers will have process insights like never before and be able to control their process based on real-time analytics. The obvious issues with traditional UV methods include the assay time. This can take anywhere from 30 minutes to 3 hours depending on how many samples you're testing. This is certainly labor-intensive time as well. We're talking about sample prep, dilutions, standard curves, and calculations. If you send samples internally to a different lab for testing, or have to wait on those results before you can proceed with further processing, these testings have to be sent to QC labs for test results. This is also time that you have to wait for these additional results. Sometimes there could be a long queue of samples sent to QC to test, or there are shifts in personnel or sample batches that you have to work around. But nevertheless, these are the bottlenecks you have to deal with before you can get your real results in order to make real-time decisions to proceed with your next step, either in PD or manufacturing. Manual dilutions and calculations can certainly add more time and error to a validation process, thus delaying the implementation process altogether and adding more time and stress to the entire process. Dilution errors can increase significantly depending on whether you are diluting volumetrically or gravimetrically. And these errors can further be compounded and cumulative and can range from 5 to 20%. As previously mentioned, sample viscosity, prep-to-prep variation, different skill sets, scale calibration, time-consuming, error-prone methods all lead to dilution error, volumetric error within the given process. The SoloVPE is the first and only at-line process analytics concentration measurement system that uses slope spectroscopy. To be very clear, this is still a UV-based method, but it is an updated and modern approach to protein measurements using VPE technology. The results require no estimations, no dilutions, and no manual calculations. Having a slope-based technology allows us to have more accuracy, better repeatability, and better data quality, which in turn allow us to have easier validation and better implementation experience with easy inter and inter-transfer of methods. Our variable Pathlength technology is adaptable to inline analytics as well. Later in this presentation, I will talk briefly about the SoloVPE system, which is our inline system that can be incorporated directly into your process for true inline monitoring for process analytics. So what is variable Pathlength spectroscopy? Traditional UV is based on Beer-Lambert's law, where absorbance is equal to extinction coefficient times pathlength its concentration. In traditional uv-vis methods, the pathlength is fixed. You have to use a standard fixed pathlength cuvette, and because your pathlength is fixed, the odds that you will have a limited concentration range available to you will need to perform dilutions on your sample to fit it within the linear range of the spectrophotometer being used. So your concentration is not fixed, it's the variable. Variable Pathlength spectroscopy is still a uv-vis method. It is also based on Beer-Lambert law. However, in variable Pathlength spectroscopy, your pathlength is not fixed. Your pathlength is the variable. Because the pathlength is variable, you have a much broader dynamic range in concentration. Therefore, you do not need to perform any dilutions, so your concentration is locked. We have variable pathlength technology, where the pathlength is the distance between the bottom of the fibrette where the light admits and the bottom of your sample vessel. In the next few slides, I will show you exactly how this system works and operates to generate data. Each sample is measured using 10 different pathlengths of data. They measure the absorbance, and the system automatically makes a linear regression curve using these 10 data points. This provides you with a slope regression. If you were to have some background interference, for example, due to the color of the medium, you would see a shift in the linear regression in the slope. However, the slope itself should be the same. Therefore, there's no interference from the medium, nor the buffer correction required. The difference between the picture above and this one with the SoloVPE system in the lower part is you having piped a connection to a system, and the fibrette is a stainless internal fiber optic that moves up and down. And you can change the path length while running a process. In 5 seconds, you can get full data acquisition of your concentration. Throughout your entire process, you can continually get concentration measurements monitoring what's going on within that given process. The system will give you the R-squared value for every slope regression, ensuring that you have linear data. The fastest acquisition in our inline technology is 5 path lengths every 5 seconds. The typical acquisition time in the at-line technology is about 1 minute for the entire measurement, again requiring no dilutions and no baseline correction. The way our technology works is by using the Agilent Carry 60 as its light source. Using a fiber optic cable, we bring the light from the Carry spectrophotometer into the SoloVPE. In this example, we are only using the Carry 60 as the light source of the device. All measurements will occur in the SoloVPE device itself. Once the user presses Start Collect, a fiber optic component called a fibrette will lower itself into the sample. This fiber is where the light emits. Path length is determined by the bottom of the vessel to where the light emits in the fibrette. Software is intuitive. It will begin to scan small path lengths to large path lengths, monitoring the change of absorbance to find one absorbance. Once it reaches the one absorbance target threshold, it will collect 10 data points of regression data to calculate your slope regression. As mentioned previously, each data acquisition takes 1 minute. So as you can see on the right-hand side, the results are instantaneous. Your slope is your OD value moving forward for all of your method development and validation for particular products using our technology. The validity of this data is based on the R-squared value, noting a pass-fail criteria of 3 nines or higher for every R-squared value. Simply dividing our slope by the product's extinction coefficient will calculate your concentration. To summarize, the concentration doesn't need adjustment. We are allowing the device to alter the path length of choice from 5 microns to 15 millimeters of path length range in as small as a 5 micron step. Whether you're plotting slope data using a regression or taking full scans by changing path length represented in the graph below, you have full visibility into your product and can successfully develop methods based on this new data acquired set that typically was not visible in the past. Here are some examples of scans using slope spectroscopy, ranging from proteins to hemoglobin analysis, gold particle viral filtration, plasmid DNA, and Listerine, showing the ability to scan over a wide wavelength range and also focusing in on the chromophores that are active within each samples to begin your method development around. A key takeaway from this information is that multiple dilutions are not required to identify each spectral scan of interest. Simply by taking this data, you can build methods around the wavelengths of interest you would calculate your slope regressions for. A few years ago, slope spectroscopy was introduced into the ASTM standard for general techniques of uv-vis quantitative analysis. This standard now contains the verbiage, to avoid the dilution step, the instrument may contain an automatic system which will allow adjustment of the path length of the measurement cell to optimize the measured absorbance. At this point in the presentation, I'm going to cover several applications and provide evidence, publications, and data within the industry to support each of these applications. As we look to the future, software plays a large role in the development and validation of slope-based methods for our customers. We have released our Viper Analytics software, which is a web-based platform which allows for specific applications to be tailored to the software for a better overall user experience. It's a very user-friendly, intuitive interface with our best practices built in to each application. LDAP ensures data integrity through managing our information, access controls, and recording traceability. Seamless integration capabilities related to SQL Server database storage and electronic data storage, and the ability to be able to validate methods in a robust 21 Part 11 environment. First, I'm going to talk about protein concentration. Using our famous quick-slope algorithm, as mentioned previously before on the slides, this is the program that's going to automate the acquisition of data. As previously mentioned, push-button analytics. There's no user intervention other than adding a few pieces of critical information to the software. On the left side, the user would add their sample name, select the vessel type of interest, wavelength, and extinction coefficient. With those 4 pieces of information, data will be generated in less than 1 minute on the right side for instant results. A few years ago, we worked with Biogen to create a published risk assessment and cost savings justification piece for the industry. Biogen had been using our technology for many years, and we wanted to highlight the overall ease of use and time savings associated with moving away from traditional UV to slope spectroscopy. Biogen proved that they were able to save a total of 59 days annually compared to their traditional UV method. System payback is very, very quick, and you can see on the left-hand side the dollar savings annually by what they save just with one system. Obviously, platforming our technology across multiple sites just multiplies the advantages, cost savings, and time savings throughout an organization. This is our most famous publication to date. Bristol Myers detailed their entire validation summary in this publication, detailing the test and overall time savings with repeatability and accuracy results well under 2%. Just like Biogen, they were able to save similar amounts of times and number of days. What is not in this paper is they were able to fit in an extra lot of material at the end of the first year of implementation. Switching gears, I'm going to talk about the use of slope spectroscopy for antibody-drug conjugates. Antibody-drug conjugates has been a problematic method for UV analysis for some time. Due to it being a hazardous material, multiple serial dilutions, and being able to measure the drug and linker at the same time proved problematic. In our technique, without any sample manipulation, we're able to look at both areas of interest using slope spectroscopy and configure the calculation for the drug-antibody ratio directly into the software. This can now potentially be set up as a validated method for this particular product with minimal sample intervention. Our Viper platform of software has completely automated this application. Similar to QuickSlope, the user would input the method criteria on the left side of the software, and you will have your concentration and DAR results on the right in less than a minute. This process makes it seamless to integrate and validate with the eventual plan of transferring these methods to partner companies or contract manufacturers who can use this and minimize the risk involved with this particular application. Some successes using the SoloVPE for antibody-drug conjugates include AbbVie in North Chicago. They looked at this method to move away from their traditional UV method and the problems it causes using traditional UV with dilutions. The high repeatability was less than 2% for them in a set of 3 different concentrations. Excellent linearity with the 3 nines criteria over for the same, and having a robust method where it enabled quick results without the need for dilution and sample prep. We then leveraged the success to work with a company who performed a full validation of the method, which is Peermill, based out of the UK. Peermill put together a publication which shows the complete validation and test results based on implementing SoloVPE for replacing their traditional UV process. In this case, they were also trying to move away from HPLC as well. Having great success offered them a cost savings of 50% compared to their old method and confirmed the use across the platforming of the company. This technology will now be used for 9 new additional products. The next application I'm going to talk about is mRNA concentration. mRNA concentration is certainly a hot topic within the industry. Our approach is using the same methodology previously used for protein concentration with multi-wavelength capabilities. We would be looking at the slope at 260 nanometers divided by the slope at 280 nanometers to account for the mRNA and the protein. You would then divide this ratio by an extinction coefficient to calculate your concentration. The same minus extinction coefficient can be used for plasmid purity as well. The difference should not be any different than the traditional method compared to, with the exception that we're removing all dilution steps and relying on the technology to establish the best linear fit within the data. This work has led to a collaboration with Aldebaran. The accuracy and repeatability performed in this study and eventual publication used 5 different levels of concentrations with 3 results per level, which exceeded the ICHQ2 recommendations. A single mRNA molecule formulated into 3 was the representative across sample matrices in order to bridge the previous study over to slope spectroscopy. The results demonstrated the capabilities of the SoloVPE technology, and the Solo is now the platform analytical method within Aldebaran's organization. This slide is just a closer look into the details of the data, showing how accurate and repeatable it was. Even though Aldebaran's criteria is set aggressively high, the SoloVPE results were nowhere near those values. Next up, flow spectroscopy for AAV titer concentrations. Leveraging the power of slow spectroscopy and its dynamic path length range, we've turned our attention to AAV viral titer. The fundamentals of data collection, again, are no different from any of the other applications and techniques I've shown. In this instance, the true power of the Viper analytics software comes into play by being able to calculate your capsid titer, genome titer, and percent full all in less than 1 minute without any sample prep and dilution error. A significant amount of work and effort went into the method development for this particular application. We leverage a very well-known Somers paper that's been in the industry for multiple years that has determined that there's virtually no change of extinction coefficient for the capsid portion of the method. We then take these published values and insert them into the software for the extinction coefficient of the capsid. We then provide a DNA calculator where users can enter their DNA sequence and have the software calculate extinction coefficients to fill in the second part of the equation for the extinction coefficient DNA. Once these values are inputted, in less than 1 minute, you will have generated results for titer, for genome, titer for capsid, and the percent full. Additionally, you'll have your R value, which is the ratio of the wavelengths combined. The R value allows you to potentially harmonize lot to lot. For example, if you had been post-processing 10 lots of material, you can now combine lots of similar ratio values, which will save you time and the cost of having to test so many lots. The highlight has led to a publication with PTC Therapeutics, where we took multiple serotypes over multiple processes to show, regardless of where things are in the process, as long as it's gone through at least one purification step, that you can get accurate measurements compared to the current methodologies of qPCR and ELISA. Same would go for ddPCR and ELISA as well. You can see the average differences between are well within range of the accepted criteria. Additionally, like our other collaborators, they've included validation data in the paper to provide the industry some evidence of what they've done to date. For AAV, it's important to note that Repligen Analytics isn't trying to replace any of the current methods that are available. What we're trying to do is provide very quick analytical approach when time is of the essence, where you need a very quick understanding of where you are within a given process to make real-time decisions. The last modality I'm going to talk about today is optical density 600. We're talking about E. coli testing over time. This application is truly meant for our inline technology. The current methods used typically involve a fixed path length sensor. The fixed path length sensor is only able of capturing a very small window into your process. The sample is either too dilute, where the sensor cannot detect the active, or too concentrated, where the sensor will saturate. Our technology gives you the full view of concentration, regardless of time and regardless of how low or high the signal is in the tank. By configuring the software, we are able to select critical data points within the process to monitor the E. coli growth over time. Older methods, which include pulling samples from the tank and measuring them offline, include issues such as continued cell growth from the time of pulling the sample to the time of measurement after dilution. The obvious issue with that is you never really know where you are within a given process. We completed a study with the Biofactory Competence Center in Switzerland that has generated a nice poster and publication related to how to measure E. coli over time using both our SoloVPE at-line technology and our FlowVPX inline technology. The paper details the test experiments, what they've done to collect the data, and the subsequent results. This is a quick summary of the data collected, comparing the FlowVPX to the SoloVPE to their former OD600, which was a fixed path-length spectroscopy method. Using Repligen analytics, you have walk-away automation that requires no user intervention or sample manipulation or chasing growth values over time. You have a truly automated process that can be walk-away. So I'm going to give you some examples of our customer successes and how, most importantly, we help. A critical note that I can't stress enough is just because you've purchased one of our systems and analytical solutions doesn't mean this is done. This is a partnership that we build with the customer to help them ensure they have success implementing our technology. It's a long journey to get towards a validated method, and it's critical that everyone understands that you're going to have our full support in doing so. We've had a very broad adoption of all VPT technology across pharma. As you can see, not only are these some of the world's largest pharmas in the world, but these are some of our largest customers that have platformed Repligen variable path length analytics across their organizations, contract manufacturing, and other partner companies. Our analytics provide a unique offering to standardize on evolved technology, which gives the users quicker turnaround, less error-prone issues, and provides real-time savings. Repligen has a plan for single alignment across the entire process, from cell culture to harvest to purification, formulation to fill finish, and then functional alignment between quality control and manufacturing. The idea to implement our technology is one process and remove all human error-prone issues related to those processes, give us the best chance of success with highlighting our analytics and providing a better window into the future. Our goal is to increase process understanding and process development from stage to acceleration and product to market. We want to reduce cycling time and increase our process efficiency, yield, throughput, resource utilization by eliminating delays associated with our traditional offline testing. We want to strengthen your process control with high quality and highly reproducible results, and most importantly, minimize the risk with continuous monitoring throughout the entire process. Our collaboration support involves many responsibilities, all of them shared. It is certainly not up to any potential customer to think they have to implement our technology on their own without any of our assistance. Our goal is to set expectations up front and early to let you know what kind of support and offerings we can help with to expedite the development and validation of methods moving forward. We will assist in your protocol development, whether it be SOPs or validation protocols, and most importantly, we will expedite these efforts to make sure that they're done correctly and have leverage throughout the entire organization that everyone can benefit from. Thank you all for attending the webinar. We'll now go to the question portion.
Thank you for a great presentation, Joe. We have a few minutes left for questions. A reminder to our audience, you can submit your questions in the Q&A box at the bottom of the screen, and any questions that we don't get to today will be answered by email after today's event. Okay, so let's get to our first question. Will future versions of a benchtop SoloVPE for protein measurement be equipped with UV LEDs as light sources, such as the FlowVPX? The LED would improve the sensitivity and also reduce the footprint.
Yes, short answer, that is actually a released product line for inline analytics. So it is now the choice of the customer to select a LED light source versus the current Carry 60 module. The main difference is the LED is single wavelength only, with a few options of choices, so it's not scanning. But yes, if you are only interested in a selective few wavelengths of interest, you can reduce the footprint of the system and go with the LED option. As far as the LED option for the at-line analytics, that is a project in the works.
Okay, terrific. Next question. Can you adjust the number of point replicates when generating the slope?
Yes to both. So you can define the number of data points that you would like to collect in the regression. The minimum is 5 data points. That's hard-coded into the software. And you can also decide on the number of replicates you would want done within a given measurement as well.
Okay. Is there any custom setting to adjust the path length increment in the software?
Yes, there is. There are 2 ways to operate the system. One is completely automated, where we rely on the software to intuitively monitor the change of absorbance while it's scanning the sample, and it will decide the path length range and path length step to use while collecting data. There's also a manual mode that's enabled where you can define the exact path lengths you want the system to be looking at.
Okay. And how are matrix effects taken into consideration?
Right. So one of the biggest features of our technology is typically you don't need to blank or baseline correct. The reason being is everything is slope-based, based on change of absorbance. Most formulation buffers and matrices don't actually have change of absorbance over path length. Typically, during a validation, you're looking to measure your buffer at the same wavelength of interest as your product. We have a criteria in place for buffers that have a slope of 0.01 or less, have no statistical relevance on the sample measurement, and can be removed. What's also nice about the technology is because it's slope-based, things like scratches or fingerprints on any of the vessels, not an issue for our technology because there's no slope in your fingerprint or a scratch. So it's a very forgiving technology.
Okay. Next question. Does this mean you do away with the reference cell? So how does the system zero the effect of the solvent?
Right. So similar to my last answer that you would prove in the validation that's not needed, and you would just run the sample in its neat form and you're done. There are certain cases when you're measuring very, very dilute samples, you would want to baseline correct. We don't take away that functionality. So it's very similar to how you would do it now, where you would blank the system with your buffer and then test the sample, and you will have baseline corrected data.
Okay. Is it possible to connect SoloVPE to Carry 100, the Carry 100 instrument?
Unfortunately not. We are tied to the Carry 60. Any future light sources that we potentially collect our analytics to are certainly could be in development. The focus right now, to go off the first question, is a much scaled down size of LED that will be used in the future.
Okay. Can you explain how the FlowVPX works for inline? In other words, sampling and measurement.
Sure. So operationally, everything is exactly the same. However, the FlowVPX model is entirely 3/16 stainless and has a flow cell. The fibrette and the flow cell are one unit. So essentially you would decide which sort of size tubing or piping you need on either end of the unit, and you'd be flowing sample horizontally across the flow cell while the fibrette is changing path length and emitting the light into a detector in the bottom of the flow cell. So operationally, the same. Certainly, if you're curious to know operationally, like to see it, we have multiple videos on Repligen's website under the analytics portion that can give you a short presentation on how the system collects data.
Okay. Do you run a standard first to get the molar absorptivity?
Typically, standards that we use, we certainly run standards for our IQ/OQ services and preventative maintenance. Most GMP groups will run a NIST traceable standard as part of their process during the week or daily activity. We would recommend the same. So again, our technology is UV-based, so whatever methods or protocols you have currently within your department, you would want to mimic for your use. So we don't tie you into any specific standard. As long as it's NIST traceable, then you would just implement comparing concentration to concentration, or our slope value to the reported absorbance multiplied by path length used to get the correlation on the certificate of certification.
Okay. And what is the minimum path length again?
The minimum path length step size for the SoloVPE is 5 microns, up to 15 millimeters, and as small as a 5 micron step. The FlowVPX has the capabilities of measuring 1 micron steps up to 5 millimeters of path length.
Okay. Can it measure all cell growth or just the E. coli that we saw in the example?
As long as there's an active chromophore for UV analysis, you can apply our technique to any UV-based method.
Okay. Can you explain how SoloVPE works for mRNA?
Sure. Certainly, all the articles and papers will be available for download and reading. For mRNA, we are taking the ratio of both wavelengths of interest, and that combined with the extinction coefficient of the product will calculate concentration. So what's nice about this particular application is in some cases the mRNA contribution and the protein contribution aren't exactly linear to each other. One might have a higher or smaller concentration contributing to the sample that may require a separate dilution. Again, because the system works off determining what's the best linear fit for data for every wavelength measured. This would just be taking the ratio of those two values and then dividing that by the extinction coefficient.
Okay. How can the validation of the process be done? Is it specific to any particular wavelength?
So we will certainly help with the validation. This, again, you can certainly start by using the current UV validation you previously used for your current method. We would obviously just work in the slope spectroscopy portion to see what's applicable and not. It is not tied to a particular wavelength on our side. It would certainly be tied to the wavelengths of interest based on your products. So we would want to base the validation study and the work around the wavelength of interest, and then design your accuracy, repeatability, linearity, intermediate precision testing, and so on based on your product.
Okay. You mentioned use of the Solo for PS80. Are there any references or application notes?
Yes, we have a few on our website that certainly can be posted here. The short takeaway of using our device for PS80 is there is a natural chromophore in PS80 at 235 nanometers. That's typically so concentrated you don't see it when you're measuring any sample with a 10 millimeter cuvette because you've already saturated. So usually the method for PS80 is in two parts, where users can now quantify working standards and batches of PS80 based off the slope value. And if you're establishing concentrations for that, you now know exactly how much PS80 you're putting into your sample based off the validity of the R-squared value and ODs measured using the Solo.
Okay. Can I use this method in conventional UV versus spectroscopy?
For comparison, I'm assuming that you mean if you're comparing, and the answer would be yes. If you're comparing slope versus absorbance, you would just want to multiply the slope value from our technology by the path length used in your traditional conventional method. That gets you the correlation. If you're going to compare it to, say, current dilution method, we always recommend measuring the same diluted sample back on our technology because we all know that the error associated with dilution error can vary between analyst to analyst. So we don't want to just back calculate by the dilution factor because that assumes that the prep was done 100% correctly. So we always try to remeasure the diluted sample as part of the experiment to correlate the values.
Okay. Is there an auto sampler for offline analysis?
Not yet. Honestly, there's no real time frame for a high throughput version of this. I would say our focus is much towards inline analytics for bioprocessing and leveraging the Repligen product lines for downstream. So short answer is no, there's no plans. However, the ability to measure neat samples without dilution or buffer correction in less than 1 minute per sample gives us a really good sort of compromise to high throughput.
Okay. Next up, is the Pathlength variation built in automatically to read data since concentration is constant in this technology?
Yes. Yes,
okay.
It is.
If I do not know the E for an unknown protein or sample, can you use this technology?
Yes, you can. It's definitely one of our really popular applications where if you know the chromophore wavelength of interest and your expected concentration, we have a feature in the software that allows you to essentially flip the Beer-Lambert law equation around that we will then be able to generate slope-based extinction coefficients based on your product's neat form. And then you can prove the validity of that value by doing a simple assay, 5 samples down to prove that that value is accurate.
Okay. Can you use SoloVPE instead of qPCR ELISA for titer measurement for release and stability testing?
Yeah, I think guys meant titer. Yes, you can. In full transparency, that's not how we've positioned the technology. There's certainly nothing stopping you. All of our data certainly has pointed us in the direction that we are comparable to qPCR or ddPCR and ELISA. Right now, we're not, as I mentioned in the presentation, we're not trying to replace those methods. We know that some of these methods are sort of the resident gold standard here, but certainly we have never run into an issue not comparing with these techniques. So individually would be a decision based from on your side to understand whether or not to move forward into sort of GMP release for this application.
Okay. Can you provide more information on the polysorbate solution analysis you listed as one of the system applications?
Yeah, I would say first check the 2 publications that we have on our website to get started, and then certainly you can reach out to me afterwards through email, and I can be more than happy to set up a time to chat with you and answer any additional questions on that.
Okay. What software do you use to find extinction coefficient for mRNA containing certain percentages of modified nucleotides? Or do you have any correction features in your software?
I would say that's a no for that one. I would need to know a little bit more about the product. Where I can tell you is absolutely a limitation of our technology, and I would make the statement that it's really a limitation of UV, is that it's not a selective analytic technique. So if there's any overlap of products or contaminants that share the same wavelength of interest, you need a separation technique. So unfortunately, if there's any method that involves overlapping chromophores at the same wavelength of interest, we're probably not the best solution. I would think UV is probably not the best solution for you.
Okay. And you highlighted collaborations with larger pharma and biotech companies. Does Repligen work with earlier stage, small, fewer than 10 full-time equivalent companies? And does the engagements that you described with the larger companies mirror what things could look like for a smaller company?
Yeah, absolutely. So I will tell you, and I promise this is the only kind of sales-related statement I'll make on the presentation. 80% of our business is in GMP environments, and it's because the adoption of slope spectroscopy as a validated release method for in-process clinical and commercial manufacturing and QC has then extended down to these type of companies that you're talking about. Our relationships with these companies are just as strong as sort of the big pharma companies. We are intimately involved with all of them and helping with their levels of development and transfers that come in as well.
Okay, thank you. A few more questions here. Lots of questions pouring in. And remember, audience, any that we don't get to will be answered by email afterwards. And also, several people have asked if the presentation is going to be available. It will be archived on CNN Online, and I think that the references will be there, available there as well. Okay, next question. Would a protein-free system suitability test be possible to verify the photometric accuracy in the UV region?
We perform those tests as part of IQOQ and PM services.
Okay. Can this be used on small molecules?
Yes, absolutely. The challenge with small molecules sort of relates back to 2 questions ago, where the good news is, yes, you can do it. Even better news that the product expected concentrations have been validated for some time. So you would just use one of your released products as your known, and then again generate your slope-based extinction coefficient based off the OD we give you and your expected concentration, and then you could apply that to the molecule method going forward.
Okay. Is 100 microliters of the sample the very minimum volume needed for analysis, or can it be lower?
It can be lower. We use a safety net of 100 microliters just because everything is dependent on pathlengths with our system, meaning the higher the concentration of sample, the smaller the pathlength used, and that equates to a lower volume. Opposite way around, a more dilute sample requires larger volumes because we need to allow our technology to use the longer pathlengths available. I would say a good sort of rule of thumb for, I'll give you a benchmark of, any MAB, that's 20, 30 mg per mil and higher, you could probably get away with 20 to 30 microliters of sample. Really, I stay away from just giving you one absolute number, but that could all be defined as part of the method development. If you were truly sample limited and we needed to revise that, you would work with one of our application specialists, and we can certainly help you with that. A key also feature of the system is that it's a non-destructive technology, so you don't actually lose your sample. We're just literally putting it in a vessel or a flow cell, so if you need it back, you take it back.
Okay. Can you distinguish between aggregated versus good protein sample?
Oh. If the aggregation is related to a typical scatter correction method that is looking to understand the presence of that aggregate related to scatter correction algorithm, the answer is yes. Outside that, I would probably need a bit more information.
Okay. What factors could cause the error in my results for SoloVPE?
Yeah, so I'll say certainly what factors could cause error, or certainly dilution error and limitations of fixed path length UV are absolutely number one and number two. As far as what can go wrong on the Solo, there are primarily two things that we cover during training and sort of education. One, by far, is sample mixing. The sample must be homogeneous before you pipette it into the SoloVPE consumable vessel. So again, the device tells you what you have at the time of measurement. So if that sample is not properly mixed, you will certainly achieve an underestimation of concentration.
Okay.
The second part would be best practices. Especially systems in larger groups where someone assumes someone else is doing the standard maintenance on the system, which is honestly about 5 minutes a week. You want to make sure that all the best practices are being performed. We have a pretty significant knowledge base article section on our website that can go over a lot of these details and questions as far as best practices and general questions about the technology.
Okay, great. Time for just a couple more questions. How do you verify the accuracy of the path length?
Sure. So what we are guaranteeing is the path length step to step. So the tolerancing of the step motor that we use for the SoloVPE is 1 micron, and we take as small as a 5 micron step. It's the same motor that turns the grating inside the Carry 60 spectrophotometer. The validity of the R-squared value in your slope regression is what gives us the evidence that you have accurate and linear data. So it is your evidence. However, as part of our IQ-OQ, we do test reference standards both ways, where we will compare slope to the reported absorbance value. We also have a test where we will physically drive the Solo to a fixed path length that was used as the same path length selected for the standard test, and compare apples to apples that way. So the verification of a fixed path length is done during IQ-OQ or preventative maintenance, but from your day-to-day sort of operation, the validity of that R-squared value being 3 nines or higher is what you would be using going forward.
Okay. What statistics are used to determine the error bars?
So there's two ways to do it. If you're using a NIST traceable standard, when it comes to accuracy, that is the only test in my mind that if any analytics is toting accuracy, I would raise an eyebrow. You can only compare accuracy related to a NIST traceable standard. So you'd certainly want to follow what's on the certificate of compliance. On that side of it, that really handles that part. I would certainly say internally, your individual departments have certainly done their own validation on where the error bars is, whether it's typically plus or minus 5% or 10% based on your drug substance or drug product. That would have to be an internally answered question by you all. What I can tell you is twofold. One, in the history, we're on 16 years of this being a product. We have never not met or been better meeting the criteria of any company we've ever worked with. And where the technology truly shines is the repeatability aspects, where we are typically plus or minus 2% for repeatability.
Okay. Last question. Can you talk a bit about the validation support that Repligen provides?
Sure. So all post-sale support is handled by two specific groups here at Repligen, one being the support group to handle general Q&A and upcoming requalifications and IQ-OQs of the system. You will also be paired with one of our application specialists that works for my group, and they will walk you along on designing a plan for your implementation efforts for that particular system. So whether it be one system that's going to be used routinely in, say, a PD environment, or if there's full plans to validate and transfer in manufacturing or QC, or develop the methods personally with analytical development, you will have specific hand-holding from my team that will be done in a combination of efforts being remote and then on-site service and testing to help you with your validation plans.
Terrific. That's about all the time we have today. Joe, do you have any final thoughts you'd like to share?
This is more just a thank you to everyone to take time out of their day in the last hour to listen about our analytics. I know how busy you all are, and it's not lost on me what it means to sort of stop your day for opportunities like this. So to the whole team that put this together, and for all of you to attend, thank you very much for your time. And I certainly encourage all of you to reach out and ask any questions you'd like post-meeting. And also, I'm highlighting the website just because we put a lot of effort into customer collaborations to provide evidence for all of you to let you know just how fortunate we are to have an evolution of UV analytics. So the more data and evidence we can provide you all, hopefully you understand just how many applications our technology can be used for.
Great. Thank you again, Joe, for a fascinating presentation. And thank you to our participants for being a great audience, and On24 for technology and production services. And thank you, Repligen, for the sponsorship that made this interactive webcast possible. Be sure to check CNEN online for information on upcoming webinars. For CNEN webinars, I'm Catherine Dold. Goodbye.